Welcome to Biolab TV, where we explore biology and lab techniques in a simple and exciting way. A mysterious illness appears. The clock is ticking. Doctors need answers fast. But how can just a tiny drop of fluid reveal what's hidden deep inside our DNA? In this video, you'll discover how a powerful technique called PCR can help solve medical mysteries, catch criminals, and even unlock secrets from ancient history. By the end of this video, you'll be able to explain the three main steps of PCR and understand how it's used to detect viruses, analyze genes, and copy DNA in the lab. Meet Dr. Alex. She's a scientist working with only a few DNA molecules from a patient's sample. That's a problem, because she needs millions of copies to study the genes. What she needs is a DNA copy machine. Luckily, she has PCR. That stands for polymerase chain reaction, a tool that can take one tiny piece of DNA and make billions of copies in just a few hours. Think of PCR like a DNA photocopier. You take a single page, the target DNA, and make lots of exact copies again and again. That's what makes PCR so powerful. It doesn't just detect DNA, it multiplies it. PCR has three main steps. These steps repeat over and over in a cycle. Step 1, denaturation, around 95 degrees Celsius. We heat the DNA almost to boiling. This breaks the hydrogen bonds and separates the double helix into two single strands. It's like unzipping a jacket. Step 2, annealing, around 50 to 65 degrees Celsius. Now we cool things down. Short pieces of DNA, called primers, attach to specific spots on the strands. Primers are like bookmarks. They show where copying should begin. Step 3, extension, about 72 degrees Celsius. At this temperature, an enzyme called Taq polymerase becomes active. Think of it as a molecular builder. It reads the strands and adds the matching DNA bases, making a brand new copy. This cycle repeats 25 to 35 times, and with every round, the amount of DNA doubles. Soon, a single strand becomes more than 1 billion copies. PCR isn't just used in science labs. It's used in forensics to match DNA at crime scenes, in hospitals to detect viruses like COVID-19 or HIV, in ancestry testing, and even in archaeology to study ancient DNA. Basically, whenever we need to find or read DNA from a tiny sample, PCR is the tool for the job. Let's quickly review the steps. Step 1, denaturation. Heat separates the DNA strands. Step 2, annealing. Primers attach to specific spots. Step 3, extension. The enzyme builds new DNA strands. Now, a quick question. What's the name of the enzyme that builds new DNA during PCR? It's Taq polymerase, named after a special bacterium that lives in hot springs. Pretty cool, right? Have you ever seen PCR in action or used it in a lab? Share your experience in the comments. And if there's a topic you'd like us to explore next, hit the like button and let us know. Thanks for watching. Subscribe now for more easy-to-understand science. Until next time, stay curious.